Okay, well, welcome to the educational workshop on network neuroscience for OHBM. My name is Alex Fornito and I'm from Monash University in Melbourne, Australia. And today we're going to have, as part of this workshop, a series of talks covering different aspects of the use and application of network neuroscience techniques, mainly to brain imaging data. And this talk here that I'll be giving is really an introduction to some of the basic concepts of network neuroscience, and this will act as a prelude to the ideas and methods that will be discussed for the remaining talks. Now, first, I just want to try and motivate why we might want to use a network approach to understand the brain. And really, it kind of boils down to the idea that there are two fundamental principles of brain organization. Now, one is that of functional segregation or specialization, and this is not a new idea. It can be traced back to Franz Joseph Gall working in the 1700s. His idea was that you could go around measuring the bumps and protrusions on someone's skull, as this was related to the size of the underlying brain tissue. And if you did that for people from all sorts of different walks of life, you could then start to build a map of what brain areas were involved in certain character traits or abilities. And so he generated a map that looked like this, and he's got traits such as destructiveness over the ear and time in the forehead and language under the eye. And we now view this is what we call phrenology, we now view it as a pseudoscience, but the basic idea that different parts of the brain are specialized to perform different functions has really been a powerful organizing force in neuroscience ever since. So if you fast forward to 1957, we have a map like this in circulation. This was trying to synthesize the results of a large number of lesion and intraoperative mapping studies. You can see that the sophistication of the terms has increased. We've got things like praxis and intellect and complex visual memory. But the basic idea that different parts of the brain are involved in different functions is still the same. Now, with the advent of modern brain imaging, we can do this with increasing precision. And this is an example I like from a study published a few years ago, where they were able to map the brain regions responsible for processing the meaning of individual words in speech. So wherever you see a particular word, that's the brain region that is responsible for making sense of it. So we can really understand functional specialization and segregation to really quite a remarkable degree of specificity, in fact, all the way down to the level of individual neurons. But we know this is only part of the story, and that's because all of these specialized processes aren't perceived as independent phenomena, but they're blended together to give us a coherent experience of the world. And that's really what refers to the other major principle of brain organization, which is what we call functional integration. And integration is really enabled by connections between different brain regions. And really, by comparison, our understanding of integration has lagged quite far behind what we know about segregation and specialization. So this image here is from a landmark study by Dan Fallerman and David Van Essen, published in 1991, where they tried to synthesize the results from a large number of track tracing studies in the macaque. And they tried to map how different parts of the macaque visual system were connected to each other. So we've got the retina down the bottom here, the LGN, V1, all the way up through to higher order areas. You can see it's an extraordinarily interconnected and complex system. Now, a couple of years later, Francis Crick of DNA fame teamed up with the renowned neuroanatomist Edward Jones, and they wrote this essay in Nature praising the work of Fallerman and Van Essen, but also noting that we had no comparable map or understanding of connectivity in the human brain. And this was really a sorry state of affairs. They made the case that if we really want to understand brain function, that we need to know how brain areas connect to each other and communicate with each other. Now, it was an interesting time for them to be writing this because just a year earlier, we had the discovery or the first reports of the bold contrast. And actually, Crick and Jones note in their paper, oh, well, there's this new imaging technique and that might prove useful. So it was quite an auspicious time. And a few years later, we had the development of the first diffusion tractography techniques that allowed for the first time in living humans to try to reconstruct white matter fiber bundles. So If you then fast forward to 2005, the techniques had become sufficiently advanced that we had in this seminal paper by Olaf Sporns, Giulio Tononi, and Rolf Cotter, we had them calling for attempts to try to generate a map of the human connectome. Now, they define a connectome as a comprehensive structural description of the network of elements and connections forming the human brain. And so really the key thing here is that the emphasis is on a network description. So we're really thinking about the brain as a network of different elements connected by axons and synapses. But if you kind of think a little bit more, you come to realize that really we find networks at all levels of resolution or scales in the brain. So we can map networks of molecular interactions that drive synaptic signaling. At the micro scale, we can map how networks of individual neurons are connected by synapses. At the mesoscale, we can map how populations of neurons are connected by axonal fibers. And at the macro scales that we typically deal with imaging, we can map how large brain areas are connected by white matter bundles. Now, regardless of the scale and the measurement technique and the species we're looking at, we can represent this network using a single common language or framework, and that's using the tools of graph theory. And we do this by distilling the network down to its bare essentials, and that's really just a set of nodes connected by edges. Now, the nodes represent some kind of fundamental processing unit in the system. So, for example, in a neuronal network, it could be an individual neuron or a population of neurons. And the edges represent some kind of interaction between the nodes, so some aspect of structural or functional connectivity. Now, we don't just use graphs to represent brain networks. In fact, we can use graphs to represent all sorts of different types of networks found in nature. So other biological networks like protein interaction networks, or social networks, or technological networks like the World Wide Web, transportation networks. Really, any system of interlinked elements can be represented as a graph. And so that provides us with a remarkably flexible tool by kind of removing a lot of the extraneous details and distilling the system down to its essential ingredients, nodes and edges. We have this very flexible and powerful tool that provides us one framework for understanding not only different types of neuroscience data at different resolutions with different measurement techniques and different species, but also for then relating those neuroscience data to a much broader class of complex systems found in nature. And so we can start to understand how the network properties of a brain might be similar or different to other types of networks. And really a key advantage of this is it allows us to draw on the tools that have been developed in the physics of complex networks and the mathematics of graph theory to try to understand the brain. Now, a critical decision when trying to build a network model of the brain is how we go about defining our nodes and edges. And really the way we do this constrains how we then interpret any analysis that happen afterwards. So I'm going to talk a little bit about this. Now, in brain networks, in some cases, how we define our nodes might be unambiguous. So at the level of, at the microscale, you might say, okay, well, each individual neuron is considered a node. But with human brain imaging, we don't have that level of resolution, and we don't really have a gold standard for parcellating the brain into nodes. So we have to use a number of heuristics. And really what we want to do when we define our nodes is satisfy three criteria. So one is that the nodes should be spatially constrained. And that just refers to the idea that whatever makes up a node should just cover a contiguous patch of the brain. You know, we shouldn't have a node that incorporates disjointed regions of the brain. And that just aligns with our intuition that a functionally specialized population of cells is confined to a particular anatomical location. The other criterion is that the node should be intrinsically homogeneous. So whatever makes up our node should share a common function. So for example, if we're clustering voxels into a single node, those voxels should share a similar function. And the third criterion is that they should be extrinsically distinct. So the functions of one node should be able to be distinguished from the functions of another. Now, there's lots of different ways of defining nodes. Brodmann's cytoarchitectonic maps are a classic example, and these have been ported into stereotactic space using probabilistic atlases for some brain regions. Alternatively, we could use variations in chemoarchitecture or sulcal and gyral landmarks. We could parcellate the brain at random. We might want to use some kind of functional criteria, for example, the results from a functional imaging experiment. There's certainly a lot of interesting data-driven methods, so trying to cluster voxels or vertices based on similarities in connectivity profiles or other properties. We could treat individual voxels as nodes, or as shown by the HCP team recently, we can use multimodal information and combine those to generate some kind of consensus parcellation. Now, I'm not going to say that any single approach is better than another. It really depends on the questions that you're trying to address and what you're interested in, and whether your node definitions actually capture the areas that you want to define. But in general, it is good practice to try to replicate your analyses or findings using different parcellations, just to ensure that your results aren't driven by the idiosyncrasies of any specific parcellation method. Now, when we define network edges, we're really talking about trying to map brain connectivity, and we can broadly distinguish between three different types of brain connectivity. So there's structural connectivity, which is the anatomical physical connections between brain regions. Now, this is intrinsically directed, so each axon has a source and a target, but we can't resolve that directionality with MRI. So that's an inherent limitation of the techniques that we're using. And there's functional connectivity. So this refers to a statistical dependence between physiological signals that we acquire in different regions. So most commonly it's estimated using a correlation coefficient between activity time series in two or more regions. But it could equally be quantified using other techniques such as mutual information, coherence, and so on. It can be either directed or undirected, and the critical thing is that it's measured at the level of the actual signals that we're acquiring. And that's distinct from the third class of connectivity, which is effective connectivity. Now, effective connectivity refers to the influence that one neural system exerts over another. And so really we're trying to make inferences at the level of neuronal populations, and we are talking about causal interactions between those populations. And so this is important because if we have an indirect measure of brain activity, such as say the bold signal, then we need some kind of mapping between that measurement and the underlying neural dynamics that generate that signal. And so a classic example of this is something like dynamic causal modeling, or DCM, where we specify different models of causal interactions at the neuronal level, and then have a forward model that makes predictions about what the actual bold signals should look like. And then we can test between competing models to work out which one best reproduces our observed bold signal. And so that's the primary difference between directed functional connectivity and effective connectivity. Is directed functional connectivity is estimated at the level of the measured signal, and effective connectivity is making inferences at the neuronal level. Now, there's lots of different ways of trying to map or measure brain connectivity. A lot of those are invasive and used primarily in animal models. In humans, we typically rely on diffusion MRI for structural imaging, functional MRI for functional connectivity. Sorry, diffusion MRI for structural connectivity, functional MRI for functional connectivity. But there is increasingly a lot of work using MEG and EEG. But by far, the most common techniques are diffusion MRI and functional MRI. Now, MRI data are inherently noisy and indirect proxies of what we're actually interested in. So we typically put those data through some kind of processing pipeline. So we'll acquire our data from the scanner, put it through some kind of complex series of processing steps, and then map our connectomes. Now, the critical thing here is what happens in the middle box here, this data processing pipeline. The meat that goes into the sausage actually has quite an effect on the final results that we get. There are a lot of different options for processing our data, and we shouldn't make these choices lightly. So, for example, head motion is one problem that's attracted a lot of attention in functional MRI. As people move their heads in the scanner, that can affect the signal we acquire and then our resulting connectivity estimates. There's lots of different approaches out there for trying to deal with head motion artifacts, and they vary quite a lot in their efficacy. So, for example, this is an image from a comparison that we did of nearly 20 different motion denoising pipelines. And so the bars represent the degree of residual motion contamination after denoising was applied. And so you can see if you use fairly simple corrections, like just regressing the 6 head motion parameters or an expanded set of parameters, you get around 70% of connections still showing some residual motion contamination. Whereas if you use more sophisticated techniques that use variants of ICA-based denoising or volume censoring, you can bring that proportion right down. And this does have a practical impact on your results. So for example, if you compare connectivity differences between patients with schizophrenia and healthy controls, you find that in some pipelines you find no differences. In other pipelines you do find differences, particularly in this case when we use global signal regression. Roughly half the differences suggest connectivity is increased in patients and half show decreases in patients. But even if you compare those pipelines that show differences, those differences are in very different locations in the network. So for example, here we have two different pipelines, one using a technique called ComCore, the other using ICA Aroma, both using global signal regression. And if we map areas where connectivity is increased in patients, you find in the top, most of the differences are located near the center of the brain, whereas in the bottom, most of them are near the posterior of the brain. So this will then affect the conclusions you draw as to which networks are most affected in the disorder. Now, what we found in our analysis was that there was no single method that was the best across lots of different benchmarks. So really there's trade-offs involved. And so that really means that you need to really understand the effect that different pre-processing techniques are having on your data and your results in order to really make an informed choice and use that information to constrain how you might interpret your findings. Now, head motion is not the only problem. Head motion is not only a problem for fMRI, it's also a problem for diffusion MRI. This is some work that we've recently done looking at over 240 different pipeline combinations. So just combining different options and choices in diffusion processing pipelines to create different types of pipelines. And again, we find that these options vary quite a lot in their efficacy for mitigating motion-related contamination. The good news here, though, is that if you use the latest version of FSL's EDDI, which includes within volume motion correction and outlier replacement, regardless of what else you do to the data, that does a pretty good job in reducing motion contamination. Now, there are other problems, of course. Motion is not the only problem in connectivity mapping. In recent years, particularly in fMRI, people have become quite interested in the role of respiration. So as people lie in the scanner, they vary in terms of their rate of respiration and their depth of respiration. And this can affect the bold signal quite dramatically. So for example, here in this top plot, you see each row is a voxel and each column is a time point, and the signal intensities in each voxel are color-coded. So light is high and dark is low. So you can see that the periods almost all the voxels increase their signal intensity, followed by periods where they decrease their intensity. And this correlates quite strongly with variations in respiration depth, as shown by this top trace here. And what was shown in this article, a really lovely paper by Jonathan Power and colleagues, was that if you acquire measurements of these physiological signals and use model-based correction techniques, you can remove most of those changes, but there's still some residual effects. Whereas if you use a technique such as global signal regression, it does a reasonable job of getting rid of those. Now, global signal regression itself is quite a controversial technique. There's been a lot of debate about the pros and cons. I've listed some articles there for people that are interested. I'm not going to say that one approach is better than the others, because it really is a decision that you need to try to make based on an understanding of the literature. But you can imagine that if different studies are either using global signal regression, as in the bottom, or not, as in the top, they're looking at completely different data sets, and the connectivity estimates are going to be very different and the results are going to change. So it is very important to be mindful of these issues and to really try to understand the impact that your pre-processing choices are having on your final results. Now that said, assuming you're happy with the way you've processed your data, you parcellate the brain, you measure some aspect of structural or functional connectivity between every pair of regions, then you can represent that connectivity in the form of an adjacency matrix or equivalently a graph representation. So these two are mathematically equivalent. We think about the brain as a graph, but most of the analyses we do and apply are actually to matrices. And so I'm going to talk about that equivalence in a little bit more detail. So here we have a zoomed in example of an adjacency matrix or a connectivity matrix. In this case, it was generated using time series correlations. So it's a functional connectivity matrix. And you see that this matrix has a diagonal here. So this diagonal represents each region's connectivity with itself. So because these are correlations, by definition, the values are 1. And above the matrix, we have what is called the upper triangle. And below the matrix, we have what is called the lower triangle. And these off-diagonal elements encode the connectivity between different pairs of regions. So here, if we go to row 1, column 2, that's the connectivity between regions 1 and 2. It's quite high, 0.8. We look at connectivity between regions 1 and 3, it's quite low, 0.45, and so on. Now, because we've measured connectivity using the correlation, this is an undirected connectivity estimate, the upper and lower triangles are symmetric. They're mirror images of each other. So if I go to row 1, column 2, I get the same value as if I go to row 2, column 1. Right? So they're mirror images of each other. And in general, we say for an undirected network, the connectivity matrix is symmetric, right? So more generally, Aij equals Aji, where the first index is the row and the second index is the column. In a directed network, so say we've mapped connectivity using DCM or track tracing or something like that, then the upper and lower triangles are not necessarily the same. So the connectivity matrix is asymmetric, or more generally, Aij does not equal Aji. And that allows us to resolve directionality. And people use different conventions. Some people will list outgoing connections along the columns, some people along the rows. So just to explore that a little bit more, here's a simple binary undirected network. So we've got the graph at the top and the corresponding matrix at the bottom. So for example, here you can see that node A connects to nodes E and C. So if we go to row A, column C, we have a connection, and column E, we have a connection. And similarly, if we go to column A, row C, and row E, we have connections. So again, the upper triangle is a mirror image of the lower triangle. The matrix is symmetric. Let's say we want to represent connectivity strength. We can do that by looking at variations in the thickness of the edges graphically, or we could just use colors to represent these changes in the matrix. So for example, here you can see a strong connection strength between nodes C and F. So if we go to row C, column F, we see a strong connectivity weight there. Compare that to the thin connection here, a weak connection between E and D. We go to row E, column D, we have a weak connection. Now here we've got a binary directed network. And in this case, you can see that A has a connection running from E. It also has one running from C and a reciprocal connection running from C back to A. In this matrix here, we list outgoing connections down the columns. So if I look at column A, I can see it has an outgoing connection to C and an outgoing connection to E, as we see in the graph. Whereas if I look at A's incoming connection, so I look at row A, there's only one connection that comes from C, which is this one here, and there's nothing from E because E doesn't send a connection back to A, right? So in this case, you can see that the upper and lower triangles are not identical, so the matrix is not symmetric, and that encodes the directionality of connectivity with, in this case, outgoing connections listed along the rows. And similarly, we can extend this to weighted networks, again, by using variations in edge thickness and colors. Given these options, you might say, okay, well then what type of network should I use to represent the brain? Well, the simplest possible representation is a binary undirected network where we just map which pairs of nodes are connected. But we know that connections do vary in strength, in some cases over orders of magnitude, so we'd want to represent that in some way. So we might say, okay, we should use a weighted network. I've also mentioned that connectivity is intrinsically directed, so we would want to represent directionality as well. Now, one of the common things of the top three graphs is that all the nodes are treated the same. There's no differences between them, but we do know that different brain regions vary in terms of their intrinsic cellular architecture, their patterns of gene expression, their intrinsic dynamics. And so we might want to represent that in some way as well, because that presumably shapes network organization and dynamics. We also know that connections are not all the same. Some are inhibitory, some are excitatory, and so on. So we want to capture that as well. So really what we're talking about, some kind of hybrid that captures node and edge heterogeneity, while also capturing weighted connectivity and directionality. And we want a network that varies through time. So functionally, on the scale of milliseconds to minutes, and structurally on the scale of days, weeks, months, and years. Now, most studies use the simplest possible representation, just a binary undirected graph. Increasingly, there's people looking at weighted graphs, and there are some studies in humans looking at directed graphs, although there is a lot more work in animal studies as we're starting to see animal connectomes be generated with track tracing and other techniques. There is some preliminary work looking at edge heterogeneity, and there's also some modeling work starting to look at the effects of node heterogeneity, but it is quite early days yet. So the nice thing about this graph theoretic or network approach is that it does give us room to grow in terms of the sophistication of our models. So as the precision of our measurements improves, we have room to grow to develop more complex, sophisticated models using this graph theoretic framework. But assuming we're happy with our graph model, we can then do two broad classes of analysis on the network. And so that's one is looking at connectivity, so just looking at variations in the strength and type of connectivity between brain regions. And the other is looking at topology. And so topological analyses are really where we try to understand how connections are organized or arranged with respect to each other. This is where we really use the mathematics of graph theory to derive a number of different quantities that capture different aspects of how the network is organized. And so really for the remainder of the workshop, the other talks are going to talk about different aspects of these types of analyses. So first up will be Janine Bijsterbosch, who's going to talk in a little bit more detail about issues associated with defining network nodes. And Shella Keilholz and Rob Smith will talk about defining functional connectivity and structural connectivity. And then in terms of analysis, Andrew Zaleski will talk about statistical inference on networks and comparing different aspects of connectivity. Bratislav Misic will talk about measures of communication and spreading on networks. Rick Betzel will talk about modularity, so understanding how different nodes cluster together in terms of their connectivity. And Sizemore Blevins will talk about multi-layer networks, which provide a framework for looking at the dynamic evolution of network organization through time or comparing different networks with similar structure. Martin van den Heuvel will talk about measures of centrality and hubs and trying to link data to other multimodal information. And finally, Petra Ritter will talk about models of dynamics that we can simulate on the connectome. So we can use connectome data to simulate models of dynamics and generate these simulations of whole brain activity. And she'll talk about how that can be done and the sort of insights that we can generate from those kind of modeling approaches. So in summary, hopefully I've convinced you that graphs can provide useful models of brain networks. They really provide us with a common language for thinking about different kinds of neuroscience data and also different kinds of network data more generally. There's different ways of defining nodes and edges for brain networks, each of which has its pros and cons. And so it's really important to keep those in mind when interpreting your findings. Brain networks can be either directed or undirected, weighted or unweighted. And there's also room to accommodate variations in or heterogeneity in nodes and edges. And we can do our analyses at the level of individual edges or nodes, or subsets of nodes or edges, or at the level of the entire network. So really, we have quite a flexible toolkit for characterizing network properties at different scales of resolution. Now, this final slide just shows some resources. There's some general introductions to network science up the top, and also to the applications of network neuroscience to the brain. There are some analysis software toolkits here to help with analysis. A few years ago, Andrew Zaleski, Ed Bullmore, and I wrote a textbook on network analysis for the brain, which is available through Amazon and Elsevier. We also have an open access journal called Network Neuroscience with Olaf Sporns as editor-in-chief. So if you are active in this space, please consider submitting your work there. And my contact details are down the bottom there if anyone has any questions. Thanks for your attention.
Hi, everyone. Thank you for tuning into this talk, which is part of the OHBM2020 Educational course on fundamental concepts and methods in network neuroscience. What I'd like to do in this talk is discuss the stage of network neuroscience where we define the nodes. And to me, this is a really important question because it boils down to how do we best represent the human brain? And what I mean with that is that we typically acquire a very complex and high-dimensional data set. I'm personally very interested in resting state functional MRI data, but the same applies to other types of imaging data where network neuroscience methods get used. And the goal of network neuroscience is really to represent the information that is present in the data that we've acquired as a number of nodes and edges that meaningfully capture brain organization and connectivity. And so the first stage of subdividing the brain up into a number of spatial nodes really forms the building block or the scaffolding for the rest of the network neuroscience analysis. So before we get started, I want to clarify some of the different concepts that I'll be using and the different words that I'll be using. So starting off, when I'm referring to a node, what I really mean is a spatially defined part of the brain that we can consider a fundamental processing unit. Another word for this really is a parcel. When we define a set of nodes, we can refer to that as defining a brain parcellation, and by extension, a parcel is one node within that parcellation. Another concept that is closely related is the idea of a region of interest, which is used in some subdisciplines of the field. And a region of interest is again very similar to a node or a parcel, although in areas where this nomenclature is used, it might be the case that there is a limited set of regions of interest that don't cover the whole brain, whereas often in network neuroscience, we have enough nodes or parcels that cover the entirety of the brain. And lastly, a related concept is the idea of a cortical area. However, a cortical area benefits from having a much more specific and clear definition. So a cortical area is a region of cortex that is distinct from neighboring cortex in terms of its function, cytoarchitecture, connectivity, or topographic organization. And that means that a node, a cortical area is likely also can be considered as a node, but a node doesn't necessarily meet all of the specific definitions of a cortical area. And things get a little bit more complex when we start talking about networks. Intuitively, the idea of a network is to refer to a collection of things, but in our field, the word network gets used to mean different things, and I'll list those just to clarify that and make it very explicit. So for starters, a network might be used to refer to all of the nodes, all of the combined nodes and edges of the brain, in the way that we refer to network neuroscience. Alternatively, a network might be used to refer to just a subset of the nodes. So for example, you can use hierarchical clustering analysis to group nodes together based on their similarity. That's also known as a module. And somewhat related to that, the last definition of networks that I'm aware of in our field is in reference to these macro-scale systems of the brain. So for example, resting state networks such as the default mode network and the salience network. And what I'd like to do in this talk, I've split this talk up in two different parts. So in the first part, I would like to cover some main considerations when we're talking about how to define and how to think about nodes. And those are listed here. And then in the second part, I'm going to talk more about challenges that we have to address when we're defining nodes. So starting off, when we think about how to define a node is to look at the modality that we're working with. So it's really important to match the way that the information that gets used to define the nodes to the modality that we want to do the subsequent network neuroscience analysis in. So for example, if we're interested in structural data, then there's a lot of atlases available based on myelination or folding or cytoarchitecture information. However, if we want to do a task analysis using task MRI, then it makes sense to delineate the boundaries of our nodes based on the activity patterns that took place in that task. And so we might use contrast analyses between different conditions that were part of the task to define the nodes. And lastly, if we're interested in resting state functional connectivity, then we really need to define our nodes based on connectivity information, either using the same data that we're subsequently going to be using for the network neuroscience analysis, or using functional atlases that are available, and some of them are listed here, that are driven by connectivity information. So the first kind of important message that I want to mention here is that we shouldn't use structurally defined nodes, such as the AAL, to do a subsequent resting state network neuroscience analysis. And the reason for that is that even though in an ideal world these nodes would look the same regardless of their modality, in reality often the boundaries that get defined from structural information don't align well with the boundaries that are functionally defined. And here I'll just include one mention of a recent atlas that was defined using multimodal information. So here the node boundaries were identified based on information from myelination, folding patterns, but also task MRI and resting state fMRI. And so perhaps that is a way to combine across these different modalities. I think that's something that we have to wait and see. So I've mentioned, and this slide is in particular relevant to resting state connectivity and nodes defined for network neuroscience analyses within that field, where there's really two different options. You can either run a data-driven parcellation, and I'll mention some of the different methods that are available for doing that, using your own data, using the same data that you're subsequently going to run that network neuroscience analysis on. Or you can use one of the atlases that is available in the literature that is driven by connectivity information. And both of these options are, there's no right or wrong here. Both of these options are valid options. And so I just want to point out some of the kind of pros and cons. If you define the nodes based on your own data that you're then going to use for the rest of your analysis, then the nodes are going to be the best possible representation of that data. And that might be important, particularly if you're working with a specific population that is expected to be different from young, healthy populations that are typically used for the atlases that are out there in the literature. So, for example, if you're working with patients who might have lesions or tumors, or younger children or older adults, then it might be beneficial to use your own data to define your nodes. However, the downside of this is that it can be a little bit more challenging to interpret and compare your results to the literature, given that you're not using the same definition of the same nodes. And so the opposite is true if you use atlases available from the literature. So here I mean atlases like the one from Yale and Schaefer, and there's many very good ones available right now. Those make it easier to relate your findings back to the literature because there's many other papers that use those atlases, but they might not be the best match to your data depending on what population you're looking at. So in the next couple of slides, I'd like to discuss some of the characteristics of how a node might, what a node might actually look like. And the first distinction here is the one between contiguous nodes, with which I mean essentially blobs. So nodes that are made up of voxels that are interconnected spatially with one another and create together one region in the brain. And this is a very intuitive idea of a node that is somewhat consistent with the idea of cortical areas. Alternatively, a node might be non-contiguous in that a single node might be allowed to consist of different blobs that are separate in the brain that are not interconnected. And while this might be a little bit less intuitive, it does actually fit quite well with the hierarchical structure that we know is apparent in the brain, and actually with hemispheric symmetry as well. Another characteristic that I want to cover here is the difference between binary and weighted nodes. So nodes can be defined as being binary, which means that for any particular voxel, it is part of one node and not of any of the other nodes. And again, this is intuitive and it allows for visualization, as I'm showing at the top of the slide here, of this patchwork of different nodes. The alternative is to define nodes as being weighted. And so in that case, each individual voxel can contribute to multiple different nodes as defined by its weight. So it might contribute a little bit to one node and a little bit to another That allows for overlap because now it's not a one or nothing in terms of node membership. And it also allows for more fuzzy boundaries, which might be a good thing to account for some of the measurement error. And I'll come back to this later. So again, with many of these choices, there's not necessarily a right or wrong, but so what I want to do in this talk is more give an overview of the different considerations that are worth thinking about when we're thinking about defining our nodes. So I'll finish this first section off by talking about a couple of different families of methods that have been used to actually perform data-driven parcellation, so to define a set of nodes. And the first family of methods are clustering methods, which essentially group voxels together based on some form of similarity to create nodes. And these are methods like Ward's clustering, K-means clustering, normalized cuts. And clustering derived nodes tend to be binary, as you can see in this figure here, and they can either be contiguous or non-contiguous. So, for example, the well-known Yeo 7 and 17 maps are non-contiguous, but many of the other ones listed here form contiguous nodes. And the second kind of family of methods that I want to introduce are what I'm referring to here as linear decomposition methods. And these methods include options such as independent component analysis, dictionary learning, non-negative matrix vectorization, principal component analysis. And all of these methods, what they have in common is that they end up giving you weighted parcellations typically, that are probably mostly also non-contiguous, so combining different regions in the brain. So to finish off this first section, I just want to emphasize that the way that nodes are defined is really, really important. These are the building blocks for the rest of your network neuroscience analysis. So if the nodes are not relevant, are not accurately reflecting the data and are not fitting to address the research question that you have, then any results, regardless of how elaborate the rest of your analysis is going to be, are going to be very hard to interpret and not particularly informative. So it makes sense to spend a little bit of time figuring out what the optimal or the most suitable way of defining your nodes is. And that might not be the same thing that someone else in your lab has done previously because of a different research question for a different study. And so with that, I'll move on to part 2 of my talk, which is what I want to do here is highlight some of the challenges that we face when we think about how to define. So what I'll do is I listed a few challenges here on this slide, and I'll go through each of these and spend a slide or two explaining what I mean. In some cases, there are some suggestions in how you might deal with it, but a lot of this talk is just about providing an overview of the different considerations and aspects that are involved when just defining the nodes for network neuroscience analysis. So the first topic that I'll talk about is node heterogeneity. And at the start of my talk, I defined a node as a fundamental processing unit. And with that, typically comes the assumption that it is a homogeneous region of the brain. And that homogeneous region can be described by a single time series that accurately captures the ongoing activity of all of the different voxels or regions that are included in that node. And this assumption that is made in the majority of network neuroscience might not actually hold true for a variety of different regions. So you can see within node heterogeneity as a result of structured noise, as a result of misalignment. So for example, if you define a region at the group level, but then it isn't quite, the borders of that aren't quite aligned with the individual, then the node itself becomes a mixture of what might be two different nodes for that person. And I'll come back to that later on as well. And there might also be true heterogeneity and the underlying neural processing And here I'll just include a little shout out to a method that we're seeing much more and more of in the literature, which is the use of connectivity gradients. And what you can do there is actually look at different axes, different directions of organization within the same region of the brain. So if that's something that's of interest, then that's certainly worth looking into further. Okay, the second challenge that I want to, that's kind of related to what I've said a little bit about already, is between subject variability. There's quite a rich literature out there now over the last decade or so with really beautiful work showing intricate individual aspects of functional organization in the brain. However, when we define nodes, they're typically defined based on group data, concatenated across multiple subjects. And that's for good reason, because we need enough data to make sure that we get a non-noisy estimate of the nodes. And we also want to make sure that there's some correspondence so that we can subsequently do a group comparison of some sort where we can relate nodes across, where we can characterize differences in nodes or edges across subjects. So we want there to be a correspondence across our population of subjects that we want to study. However, when you define nodes at the group level, you're inevitably going to miss out on some of that individualized spatial organization. This has been recognized in the literature now, and there's actually quite a number of different methods available that assume a group level node organization, but then perform an extra step of estimating individualized subject-specific versions of that group organization. I've listed a few of them here. So for example, you can train a classifier based on different features to learn the node boundaries for individual subjects. You can use dual regression, which is paired with independent component analysis. You can use Perfumo, which is a method that I'm showing the figure here on the right is referring to, and this is actually a hierarchical method where the subject-specific and group-level estimates are iteratively optimized. Or you can use task-based activation mapping if you have a task that uniquely activates a node of particular interest. So moving on to the next challenge, and here I want to talk a little bit about dimensionality. So the number of nodes needed to cover the entire brain. And really there's no clear-cut best dimensionality, and most likely there's a lot of different accurate dimensionalities because the brain is organized in a really hierarchical fashion, so that you can probably characterize it, summarize the brain well, both at, using a very low set of these very large-scale networks, and also using hundreds of smaller contiguous nodes. So this is an answer where there's no real, this is a question where there's no real obvious best answer, and potentially trying multiple different dimensionalities can be informative to test if the results are consistent across that. The next thing, so I've talked already about between subject variability, so how organization might vary from one subject to another, but actually the definition of nodes themselves might vary within an individual over time. And there's a few different examples of this. This is an area that we know relatively little about, but it is possible that different states might result in a shift of the organization of the functional regions, for example, in response to task demand or emotional states. It is also possible that dynamics is something that has been studied already, but dynamics in terms of the actual organization of the nodes themselves is something that's rarely looked at. And it might actually be the case that nodes themselves reorganize in response to dynamic changes such as mind wandering and arousal, and potentially many others as well. And the last, within subject changes over a longer time scale, so longitudinal trajectories that might be related to development, aging, different stages, different disease stages. And so changes in node organization in relation to these within subject changes is really something that we might see a lot more research in coming years. The last challenge that I'd like to talk about a little bit more is the idea of representational ambiguity. And here, what I'm really talking about is in network neuroscience and in many other fields, we typically see the step where we define the nodes and then the step where we define the edges, which future talks will discuss more, as kind of separate entities, as unrelated stages. However, when we define the nodes from our own data and we then define the edges from our own data, they're both driven by the same underlying information, the same underlying data set. And therefore, there can be a lot of shared information and a lot of ambiguity in how these different entities are defined, much more so than we often take into consideration. So I just want to show a couple of examples of this very quickly. So this is the first example where the figure on the left here is showing significant edges, so edges that were significantly different between scan 1 and scan 2 in the Human Connectome Project data. And that's unexpected because these are back-to-back scans with no interesting intervention or anything taking place. So we expect the data to be very similar across these two scans, and yet we see these very significant differences. And when I used the additive signal change analysis, which is developed by Eugene Duff, we can show that all of these significant edge differences are actually caused by changes in the signal strength or the amplitude of individual nodes. And so that's showing you that an edge can, an edge estimate can be influenced by aspects that don't necessarily relate to connectivity, such as the amplitude of the signal in a certain region. Another example, and we looked into this a little bit further, here we, again in data from the Human Connectome Project, estimated individual estimates of the spatial node organization, the signal strength, and the covariance structure, or the correlation structure, or the edge structure between nodes. And then we manipulated those estimates such that we fixed some of them to the group average so that they didn't vary across subjects. So in this case, the only thing that was allowed to vary across subjects was the spatial definitions, the individualized spatial organization of the nodes. Then we obtained a simulated estimate, having manipulated the results in these ways of the subject data, that we can then use to estimate edges between the nodes. And we can ask the question now, how much of the variance of the original edges can we explain by this simulated version of the edges? And the answer to that should be none at all, because we fixed the time series such that the correlation structure, or the edge structure between the nodes, didn't vary across subjects. But actually, we found that a large proportion of the variance can be explained by just the spatial organization of the nodes. And so this is to show you that not accounting for variance, spatial variance, when defining the nodes, can actually affect the edge estimates that you subsequently get in subsequent analyses. And so here on this slide, I'll summarize some of what I see as the main takeaways, and I'm very happy to have a discussion on this, because I think there's probably other things as well, but these are some of the things that I think are important when considering node definition. So the first one here is go for data-driven nodes, either from your own data or from data that's from high-quality atlases of a matched modality that are available in the literature. The second suggestion that or takeaway that I would have is to inform the definition of your nodes, the choice of your nodes based on the modality, but also the research question and the hypothesis. So take a moment to stop and think about, given all of the considerations that I've highlighted in this talk, what nodes might be most suitable for the study that you're about to do. There is a lot of spatial variability, and so I think more and more it will be of interest to try and estimate subject-specific boundaries using some of the methods that I've highlighted, and new methods will likely start to come out as well. Given that there's no real right dimensionality, it makes sense to test results in any particular study across multiple dimensionalities. And we're seeing this already quite a bit, to see that the results are not a fluke that is specific to one dimensionality, but are pretty robust again across the range. One of the things that I'm really interested in, and that I think could be very, very valuable, is to compare in the network neuroscience analysis that you're going to do different types of parcellations or node definitions. So for example, if you define nodes using a weighted non-contiguous regions or as contiguous binary regions, does the essence of the result change in a way that might be informative, or is it actually very robust to this? When writing up the results, I think we can all, and I include myself in this, do better to be very explicit in terms of including a justification. Why is this the set of nodes and the way that nodes are defined suitable for the study that they are used in? Explicitly state assumptions, so things like that, some of the things that I've mentioned in this talk, like whether you're assuming homogeneity and other steps, I think can be very helpful to help people understand the perspective of the how to interpret the results. Avoiding overinterpretation, I think it's important to remind ourselves that we're not actually measuring neurons and axons, but we're very many steps away from that, regardless of which modality we're working in. It's good to be careful in terms of the wording that we choose for interpreting some of the results. And lastly, openly share all of your nodes, results, code. That's really the best way in which other people can build on your results. So that's really important. So with this last slide, I just want to ask a question which relates to, is there potentially a difference between the best model or the best representation of the brain versus the best model or the best representation of the data that we measure with MRI of the brain? And so that's just to keep in mind that when we gather data with MRI, whichever type of, whether it's structural or functional, we get these laughably large voxel sizes, which are huge in relation to individual neurons or cells. And in addition to that, if we're doing functional imaging, we have to remember this hemodynamic response function, which might vary across subjects, which might vary across different areas of the brain. In addition to that, our data are just very, very noisy, even after pre-processing and careful cleanup of the data. And then the last thing is that we're really looking at a very, very indirect measure of the thing that we're trying to make inferences on. And so all of this together means that the way that we conceptualize, or the way that we, in our theorizing, model the brain, doesn't necessarily map on one-on-one to the way that we best model MRI data. And so, for example, it might be that there are real hard cortical boundaries between cortical areas in the brain, but given the spatial and temporal resolution and noise factors and everything else, using a fuzzy weighted parcellation or node definition rather than a hard one might actually be a better model of MRI. And I think that's a discussion that is an interesting one to have and an important one to have for all of us. And so with that, I'll finish, and I'll thank all of my wonderful collaborators all around the world. I'll thank my lab at WashU, who have shown incredible resilience and are still absolutely fabulous even whilst we're social distancing. And I'll also thank my quarantine mates or office mates at this time right now. Thank you very much for your attention.
Hello, everyone, and welcome to the OHBM educational course on network modeling. So for this course, I've been tasked with telling you about functional connectivity methods to measure it, and metrics to characterize it. So just a quick overview. I'm going to divide the topics we're going to cover up into a few different sections. The first is average or static functional connectivity. And this is the most traditional type of functional connectivity analysis where metrics are calculated over the course of the entire scan. So for example, you would get one correlation value for two areas for the entire scan time. I mentioned correlation. That's one of the common approaches for average functional connectivity. Another is ICA, independent component analysis. And then there are several other methods that I'll just very briefly touch upon. So the other big category is going to be time-varying functional connectivity, sometimes called dynamic functional connectivity. And again, there are many, many different ways to measure this, but I've broken it down into a few different categories based on windowed methods, event-based methods, and then repeated patterns. So we'll go into kind of the generic categories that you can have for this type of analysis. And then I'm going to touch briefly on some summary metrics, how you might put this data together, and talk a little bit about null models and validation. So to begin with, we're looking at average functional connectivity. And first thing I want to bring up is the idea of whether we're looking at activity within an area or coordination across areas. And I want to make this distinction because different methods give you different answers for these. So we often think of activity within an area as being something that we look at with fMRI, where you know the timing of a task and you want to see which area becomes active. But there are also metrics that you can use with resting state fMRI that also give you some sort of estimate of area-based activity. And the examples that I put up here are the amplitude of low frequency fluctuations and the fractional amplitude of low frequency fluctuations, ALFF and FALFF, which tell you something about the activity distribution within an area in terms of frequency. Now, that said, I'm going to ignore these kind of metrics for the rest of this talk. And instead, I'm going to talk about metrics that tell you about coordination across areas, because that's more what we think about when we talk about functional connectivity. So when you're thinking about coordination metrics for resting state fMRI, this came about because the signals are low when we're talking about these spontaneous fluctuations. So there's no obvious excursion away from the mean that we can use to say, oh, something is happening here. And the timings of our task or stimuli are unknown. And I've put task and stimuli in quotation marks because, of course, for resting state fMRI, we don't really know what's going on in somebody's head. We don't know what the task is. We don't know what sort of stimuli they're receiving. So the idea behind traditional functional connectivity analysis is that you have neural activity in an area that leads to a change in metabolism, which in turn leads to a hemodynamic response, and that's what we measure with the bold signal. So we've got this happening all across the brain, and if we look at the correlation between the bold signal from two different areas, we then kind of feed that back through this chain of events and use it to, we interpret that as meaning that neural activity in those two areas is also correlated, or that there's some sort of coordination of activity going on. So just a brief aside then, because we're working backwards from this correlation that we see in the bold signal, other processes can kind of intervene in that back projection to neural activity. And so there are things like physiological cycles like respiration, the cardiac cycle, there's head motion, and all of these can introduce correlation into the bold signal that's not directly related to neural activity. So when we're interpreting that correlation, this might mistakenly be assigned to having correlated neural activity between areas, when really it's one of these midline processes, some sort of physiological noise that's contributing. I'm not going to go into that in great detail. There's been a substantial body of work on how you might characterize these sorts of noise and correct for them. Okay, so back to correlation. I'm picking correlation to talk about first because it's still the most widely used metric for functional connectivity, and it's really one of the simplest to understand. The equation is pretty straightforward. So if you look over here, I'm going to underline it. When we calculate correlation, we're just looking at how the deviation from the mean in two pairs of signals corresponds basically. So we multiply the deviation from the mean in one signal by the deviation from the mean in another signal, and then normalize by the standard deviations of the two signals. So this is a measure of linear dependence. And depending on how you set up your scan, you might calculate correlation between the time courses from different voxels, the time courses from different regions of interest, ROIs, or the time courses from different networks. So I think it's helpful to have this sort of visual depiction of what correlation can represent. And I've just pulled some images off of Wikipedia for what I'm showing here. So if we look at this first row over on the right, this is what we usually think of correlation as reflecting. We might have perfect correlation, whereas one variable goes up, the other variable goes up in a very predictable manner. As our correlation values go down, the relationship gets more noisy, a little less predictable, but you can still see trends. When you get to 0, there's no, what you know about one variable tells you nothing about the other variable. And then when we start looking at anti-correlation, now we start to get a relationship again, but now one variable goes down as the other goes up until we get to perfect anti-correlation. Now note that correlation says nothing about the strength of the relationship between the two, or I should say like the slope. So if you look at the second row, you can see that all of these are either, all of these relationships are either perfectly correlated or perfectly anti-correlated, except for the one where the line is completely flat. I also mentioned that correlation measured linear dependencies. So if you look at this bottom row right here, these are examples of structure that occur between two variables that will give you 0 correlation. So it's clear that there's some sort of structured relationship, but correlation doesn't capture it. Now I think that the figures at the bottom are even more informative. So again, in this first figure right here, we have what we kind of expect correlation to be, a nice line fit between some kind of noisy data that catches the trend of the relationship. So again, with correlation being a linear measure, if you look over here now, you can see that we actually have a nonlinear relationship that gives us the same correlation as this linear relationship that we looked at just a second ago. So it's approximating this nonlinear relationship as something linear. And then down here, you can see on this point, we have something that's perfectly correlated except for an outlier, which changes your correlation value. And then this one is my favorite, and the thing that worries me the most when I see it in a paper that someone submitted. This is correlation that's completely driven by an outlier. So here you have basically all of your values clustered in one spot and one outlier that's really making you fit that linear value. Okay, so correlation doesn't depend on the amplitude of the two signals. So here I've plotted in MATLAB a signal, one that has about twice the amplitude of the other, so that you see those in red and blue. And you can see that they're perfectly correlated, even though their amplitudes are different. But when I add noise, correlation falls down fast. So here's half a sigma Gaussian noise added, and suddenly instead of 1, the correlation coefficient is 0.6 about. When we go to even higher noise, the correlation gets even lower. So I wanted to put this up as a demonstration because a lot of times when we're talking about signals from resting state fMRI, people say, well, amplitude doesn't matter for correlation, and that's true. But signal to noise ratio does matter. And so if your signal to noise ratio is different for two signals that are perfectly correlated, you're still going to get a different answer. So there are a couple of really common correlation-based approaches for calculating average functional connectivity. One is seed-based correlation, and I'm showing that here at the top. The idea is that you take some area that you're interested in, and here it's this sort of, my pen's making a little mark there, but this orange area kind of at the front of the brain, the bright yellow. So I have a time course, here's my ROI, and I get some sort of time course from that. I take that time course, I correlate it with everything from the rest of the brain, and then at each voxel, I decide if that correlation coefficient is significant or not, and if it is, I paint it over this anatomical image. So that's how these maps are usually obtained. Another way to do this is to look at the whole brain at once. So let's say I didn't know that I wanted to look at prefrontal cortex. And instead, I take the brain and I'm going to just kind of roughly divide it up into sections. Now, this is obviously a very crude parcellation that no one would use. If you want it, I'll be happy to recreate it for you and make it freely available. Okay, but there's my brain parcellation. And now what I do is I take the average time course from each one of these parcels, and I make a matrix of the correlation between the time course of parcel 1 with the time course of parcel 2, 3, 4, 5, 6, etc. And so maybe, if I pick a point, maybe I have, this time course goes here, and the time course from this other area goes here, and the point where those row and columns intersect tells me the correlation between those two time courses. So note that this is symmetric. I could just as easily have said the second one goes over here and the first one goes over here, and I would get the same value. Okay, so going back to the seed-based correlation for a minute, I did want to show how you can get different networks if you change where you put your seed. So this is data from the same animal, and we're showing 4 different seed locations. And the first one, the seed is up here where you see this bright red spot of high correlation, that's the seed, here in somatosensory cortex, and we're seeing a network that goes across left and right S1. You can do the same thing with a seed in the hippocampus, a seed in the caudate putamen, or a seed in S2. And each of these gives you a different network. So then if you want to compare across animals or people, I know I'm using rat brains here. That might throw some of you off. I like rats a lot, and I think they have beautiful brains. But the principles are the same if you do this for humans. So anyway, if you want to compare across animals or people, you can register each image to an atlas, and that gives you a common space where then you can lay over either the seed-based correlation maps, or you can also use this sort of atlas to parcellate the brain and do a much better job than I did on the rough example that I gave you earlier. So this lets us put correlation maps or correlation matrices from different animals into the same space, or from different people. Okay, so to go back to those correlation matrices then for a minute, I mentioned that these are symmetric. There are also a lot of different kind of shapes you'll see, and it depends on how people sort the correlation matrix. So in this case, we've sorted it so that default mode networks are members are mostly here at the top, up here, and then the anti-correlated task-positive network is at the bottom. And so you see strong correlation within default mode network components, strong correlation within task-positive network components, and strong anti-correlation between the two different networks. Now, you can rearrange this in a lot of different ways, and often you'll see these sort of blocks along the diagonal that indicate kind of these modular networks. Because these are symmetric, we often show, say, a healthy group and a patient group on one side or the other of the diagonal, which is what we've done here. The bottom left is the control group, the top right is functional connectivity and a group with ADHD. And then over on the right, what we're showing is the differences between the two groups using a couple of different methods that really aren't important for this particular presentation. But now we're showing just which connections are significantly different across the groups. Okay, so one of the problems with correlation is that it gives you a distribution that's not normal. It can't be normal, right? Because the minimum correlation is -1, the maximum correlation is +1. And so before you do statistical analysis, you've got to make this look something that's more like a normal distribution. And the way people usually do that is using the Fisher transform, which is basically taking the arc hyperbolic tangent. And so that spreads out those values that are near positive and -1 and gives a wider distribution. So the other factor that you have to remember is that we need to correct for multiple comparisons. So let's say we have that matrix and we've parcellated the brain into 360 areas. So, this is something people use for the cortex when they use the Glasser parcellation for HCP data. Well, if you do 360 areas times 360 areas for all the comparisons you can make, that's over 129,000 correlation values, or differences in correlation, depending on what you're looking at. So, and I know that that's including the whole matrix. So if we did half of it, it'd be half of that. If your matrix then is set, if your threshold is set to have a significance threshold of 0.05, then that means if you multiply 0.05 times 129,600, you get 6,480 significant values just by chance. And so we have to find a way to correct for that, for these significant values that show up just by the virtue of the sheer number of the comparisons that we're doing. And there are a bunch of different strategies. I'm not going to go into those in this presentation. I just want to throw those up there so you have something to look at if you're interested in this area. Things from Bonferroni correction, where you basically divide by the number of comparisons you're making, clustering with the idea that significant values don't necessarily fall next to each other if they occur by chance, and then there's many others. Okay, let's sum up correlation then. We know that it captures linear relationships. It may or may not pick up nonlinear ones. I think it's relatively easy to understand what it means when you have a difference in correlation. I mean, at least the equations are pretty straightforward. We know that it's sensitive to changes in signal to noise level, and in practice, in resting state fMRI, these can arise from things like vascular changes, different levels of motion, drug effects, and I include caffeine in that. And these correlation values are the basis for many subsequent analysis. So you can do other things after you calculate the correlation between areas. So I like to put interpret with caution. So if you do see a difference in correlation between groups, you want to make sure that they have similar levels of motion, that the amplitudes and the frequencies of the bold signals are the same. And then global signal is another potential confound that I'm not going to really talk about today. It kind of goes back to the discussion of nuisance variables, and is something that you need to decide how you're going to handle. Okay, so the other most widely used method for average functional connectivity analysis is independent component analysis, or ICA. And the idea behind ICA is very similar to PCA, where you separate your signal, your multivariate signals based on how much variance they explain. But here we're going to separate a multivariate signal into independent non-Gaussian signals based on trying to maximize their statistical independence. It's usually applied spatially for resting state fMRI, and that means the areas that have similar signals tend to be grouped together. There are many different algorithms you can use to calculate ICA, and some depend on minimizing the mutual information between different components. Some try to maximize the non-Gaussianness of the components. But one thing to remember in terms of interpretation is that ICA doesn't inherently tell you how many components there should be or how strong they are. So that's kind of a judgment call that you have to make when you're doing analysis. And I have an example to show here. So this is ICA on some human data that my lab did. I think we used something like 30 ICA components when we were doing this analysis. And why did we pick that? Well, it was close to what people had used before. But you can see that by picking the number of components that we did, we've made choices that affect how our networks come out. Also notice first that I'm not showing all the components because a bunch of the components looked more or less like noise components. And so we got rid of those to try to clean up the signal. But okay, going back to the components that we do have, here you can see we have a component that looks like the posterior cingulate. And here you can see we have another component that looks a lot like portions of the default mode network. So if we had picked fewer components, maybe those two components would have merged to become a single component. And maybe that would have been the right thing to do, but how do you know? On the other hand, you can see that there are, in this default mode, there are things that we might reasonably consider single nodes. And so it might have also made more sense to do a higher number of components and try to have each component be a single contiguous spatial node. So there are different ways to do this, and I don't want to say that one is better than another. I think it's just a matter of what you're trying to get with your analysis and how you want to be able to interpret it. Okay, so the major advantage for ICA is that it's data-driven. So you can use it at the single subject level. More often it's used on a concatenated set of data to provide group results. You can use it as the basis for another type of analysis. For example, if you use ICA to pull out kind of single spatial nodes, then you can do correlation between those nodes. Just use the ICA as a sort of parcellation. Sometimes ICA can separate out noise components, which you can discard then to kind of clean up your signal. And there are different approaches to do that. There's fixed, there's melodic, and there's just, kind of visual interpretation of each component, trying to decide which ones look like motion, for example. ICA assumes spatial independence. It's unlikely to be true, but it still is a very useful tool for resting state fMRI. I think one of the main challenges for me, besides the idea of trying to figure out how many components you should use and how strong the components are, is that it's hard to interpret differences that you get across groups. So if you put a patient group and a healthy control group together to do the analysis, you're kind of enforcing the same spatial structure if you use basic ICA. But if you don't concatenate the two, you might get differences in the components that are really not very easy to understand. And ICA is an area of very active research, and there are a lot of more sophisticated versions out there that try to address some of these issues. Okay, so other measures of average functional connectivity. One is partial correlation. This is a method that reduces the effect of common inputs. There are a bunch of mutual information measures which can capture more of the nonlinear dependencies. There are things like coherence or partial coherence, where you look at basically correlation in the frequency domain across frequencies, which can be phase insensitive. And I think there are more methods than I can possibly go over, but for a great demonstration of how you might compare some of these metrics and how some of them stack up in handling different scenarios, you might look at this paper by Steve Smith that came out in 2011. It's really fantastic. Okay, so then moving on to time varying or dynamic functional connectivity. The difference here is that for most average analysis techniques, we get one metric of connectivity for the whole scan. So let's say one correlation map with our seed, one correlation matrix between our parcellations of our brain. For dynamics, we want to get more than one of these measures. We want to get something that varies over the time of the scan. And so you can imagine a very simple approach is to divide the data into windows and then use the same methods. So you have a 10-minute scan, maybe you break it up into 1-minute windows and you calculate your metrics for each of those windows. You can do this by just chopping up the time series. You can do this by taking a sliding window that you move along the time series, or you can use more of an adaptive window approach where you have some criteria that tells you how you want to segment your data. I'm just going to stick with sliding window methods for the purposes of this talk because they're still pretty widely used. But there's a lot of research out there that gets into different windowed approaches and also different dynamic approaches. I'm going to talk a little bit about two that are based on individual events or repeated patterns, but I have to acknowledge that this is really just giving you a flavor of the wide variety of approaches that are available. Okay, so let's talk about sliding window correlation. So let's say I've got my gray window here. That's the area of the time course I'm looking at. I've got my two seeds. I calculate one correlation value. Now I slide my window along. I have my previous correlation value at time t equal 1. And now at time t equal 2, I have another correlation value that's a little bit higher. And so you can imagine I keep repeating this and I get some sort of time course of correlation over time for these two areas. So this is what it looks like empirically. I'm showing the correlation time course from two areas that don't have significant average functional connectivity. So the average functional connectivity is around 0. But if you look at it on a windowed basis, you start to see fluctuations. And the size of the fluctuations depends on the size of the window. If you have shorter windows, here I'm showing 25 time points in the blue with the dots, you see much higher variability than you do with the long 100 time point windows shown in the black. So when we first started looking at dynamic functional connectivity with sliding windows, back in early days when people were just starting to do dynamic analysis, we were very excited to see the sort of variability between areas that didn't have strong average functional connectivity. We thought, well, maybe it's because sometimes they're working together, sometimes they're anti-correlated, and then it all averages out to 0. And so that plot that we made, if you look at that naively, it seems to support this idea. So we made plots then of how much of the time course each pair of areas spent in a high, low, or medium correlation. And so I'm going to highlight the same sort of area over here. So this is an area that had very low average functional connectivity. And we thought, wow, look, it really varies a lot. The problem comes is that if you look at areas that are randomly matched, so maybe time courses from 2 different rats, you see the same sort of variability. And that made us worry a little bit about this use of sliding window correlation, because maybe we're just picking up properties of the signal itself. So we did some network modeling, just using simulations where we knew the ground truth of the correlation for our 2 time courses, and then we looked at how well sliding window correlation could pick that up. And turns out it doesn't do very well at all. So that was kind of alarming. So if you look up at the top, the black solid lines are showing you what the correlation actually is, and the colored lines are showing you the correlation that you calculate using sliding windows for 25 TRs at the far left to long windows of 200 TRs at the far right. And none of them do very well. So that was disappointing. That said, we also looked at sliding window correlation using bold signal and electrophysiology and found that the bold values were actually quite correlated with what we saw in the electrophysiology. So it's capturing something about the underlying neural activity. And if you look, there have been behavioral studies, and if you look at how sliding window correlation predicts performance, you can actually see that it tells you something about how well people are going to perform a very simple task on an individual basis or a between individual basis, both. So that seems to suggest there's something there. So I just want to acknowledge kind of the controversy around sliding window correlation. My lab is on both sides of this particular fence. We've seen evidence that it's not very accurate, but we've also seen evidence that it's quite useful. I think maybe the performance depends on the signal to noise ratio. If you're doing comparing 2 sliding window approaches based on, say, neural activity in the bold, it might be that you distort the data in the same way. And even if we have a poor representation of true correlation, you might get some useful information out of it. So just one of those things that you should be aware of both sides of the argument. Okay, so going on then to a different type of analysis that gets away from this issue with the windows is analysis based on individual events. And there are a lot of different versions of this out there. Some of the earliest I saw came from Penny Golan's group. And basically the idea is that you find some way to identify individual events in the resting state fMRI signal. It might be by deconvolution. It might be as simple as taking the time course from an area and detecting when there's a peak. And that's what I'm showing here at the right. Now, if you do that, you know when that area is active, and you can kind of look through other areas' time courses and see which areas are also active at those times and make, patterns of co-activation patterns for areas that become active together. So this is an example from the paper by Wu and Dunn and PNAS in 2013. So up here, they've got a single frame of co-activation pattern. So this is, what happens when one of these areas, I think it was the precuneus here, goes above a threshold, this is the pattern that they see across the brain. If they average all the patterns when it goes above the threshold for a run, they get this middle row here. And then if they average over many scans, it looks like this. Now for comparison, they've shown the same maps where they've used the precuneus as a seed region over here on the right. And you can see they look very similar to the coactivation patterns. So it looks like maybe these coactivation patterns are really capturing the high amplitude events that are driving this average functional connectivity and might give us a way to look at it in a more time-resolved way. So I think that's a very cool approach. Another thing that my lab has done that it kind of extends upon this is instead of looking for repeated patterns of spatial activity, we look for repeated time course patterns of spatial activity, meaning that these patterns are 4D. So we look for dynamics that repeat over time. And I'm going to show you an example of what I mean. This is a pattern that's going on in the background of resting state functional MRI scans all the time. This time course at the top shows when it occurs, these strong peaks, or the occurrence of this pattern at different time points in the scan. Okay, so you're going to see areas where the activity is higher than its mean are in warm colors, where it's lower than its mean are in blue, cool colors. And you'll see kind of this switching between two major networks over time and also propagation along the cortex. And again, this is something I'm not going to get into in much detail today, but I wanted to point this out as a method for dynamic analysis. You know, our latest work shows that you can summarize a lot of resting state fMRI with just a few of these patterns. Okay, so the problem with time variant analysis, which I really love a lot, is that it gives you too much data. So instead of just having thousands of voxels per person, now we have thousands of voxels per person at hundreds of time points per person. So you get a data explosion. And you need to find ways to summarize and compare across different groups. So there are different models. I'm going to talk about two just briefly to give you an idea very quickly. So the first way is to maybe summarize patterns of activity into brain states. And these are very loosely defined because nobody really knows exactly what the right temporal or spatial scales for brain states are. So we usually use this to refer to patterns that repeat over the course of a scan. But how often do they repeat? How similar do they need to be? I don't think we really know. But just as a practical matter, these are often calculated by either doing sliding window correlation and then clustering the results, or doing just taking the raw activity of the brain and then clustering the results. So here are a couple of activities. Here are a couple of examples. Here we've used just clustering on the raw activity to show that you can get two major states easily at the individual level. We call this one at the top loosely the task positive network, the one at the bottom loosely the default mode network, and this time course at the bottom showing the red and the blue shows how a single individual might flip between these states during the course of a scan. And so you can calculate things like the dwell time, how long a person stays in one state, how likely they are to transition between states. So that's one extreme where we have only two states. At the other extreme, we can have maybe 7 states here, and we can do the same sorts of calculations. Now our states are defined by correlation matrices between the different brain areas, and we're looking at how those correlation matrices change over time. Same idea, just a different formulation. But I think this also highlights one of the challenges, you know, just like in independent component analysis, it's hard to know how many components to have. When you're looking at brain states, it's hard to know how many brain states you should have. And for example, if you look at these two, it kind of looks like one is just a reduced amplitude version of the other. So I don't think we have a really solid way to figure this out just yet. Okay, so briefly, I'm just going to mention, because this is network modeling, that graph theory is a great way to summarize brain activity. You choose each region of interest or voxel as a node, and you decide how strongly it's connected to other nodes using our correlation values or other metrics of functional connectivity. So these are usually weighted connections, but undirected, and they can be used for static or dynamic analysis. You're going to hear a lot more about this in this course. So I'm going to go through this pretty quickly. I've got a list here of graph theory metrics that are up there, but like I said, there are people who are much more knowledgeable than me who are going to be talking about this. So I'll refer to the further talks for that. I did want to mention that, you know, one way to think about this as a global metric is you can do something like the segregation versus the integrated state of the brain based on, you know, how much is divided up into separate modules versus how well those modules are all connected together. And you can look at that as a single metric that you can describe brain dynamics with over time. So that's kind of the extreme end of data reduction, trying to tame this data explosion that we get out of the dynamics. Again, I know that I'm running a little long here, so I want to finish up quickly. The statistics for data analysis, I think, is a very active area of research. It's difficult to find the right models sometimes. You know, we started out doing permutations and mixing data from different subjects, but that doesn't necessarily preserve the static or average functional connectivity value. So you really have to be careful about trying to match your null models to your to what you're trying to find out. Okay, so I wanted to wind up with just a couple of things. First, your consideration. So with all of this information, how do you decide what to do? And really, I think the decision has to be made on a study-by-study basis, depending on whether you have a hypothesis about certain areas being involved, or if you need whole brain information. Is average functional connectivity sufficient, or do you need to look at how things change over time? Are you interested in connectivity between regions? Or are you interested in more complex metric measures? I think it's always good to remember that there are factors that differ between groups that can influence whatever metric you're using, whether it's the amplitude of the bold fluctuation, the noise from the physiological processes or motion, or other choices that you've made during your experimental design. And of course, you need to think about how you're going to do the right statistical analysis. So the other things that I really wanted to point out is there are a lot of caveats to any of these approaches. Remember that we're looking at hemodynamics, and neurovascular coupling isn't straightforward. You know, we've done a recent study looking at electrophysiology and the bold signal in rats, and you know, you can have very high amplitude bold responses with no underlying change in the neural activity, or vice versa. Now, on average, of course, there's a relationship, and that's how we pull it out. But it's a very messy sort of thing. When you're combining data in these frameworks to look across subjects, registration and smoothing has a big impact on our ability to detect small areas. We're taking that beautiful individual level data and smearing it all over the place so that we can do group analysis. Should always consider the impact of noise, because noise can reduce your apparent values of correlation. And then the thing I think is really important is that any higher level analysis you do, for example, if you're looking at a graph theory summary that's based on correlation, whatever changes you see in modularity based on graph theory still rely on changes in that underlying correlation metric. So you need to keep those considerations for that baseline metric in mind when you're interpreting your higher level analysis. So I hope everybody will try to maintain a healthy skepticism when it comes to interpretation of resting state fMRI studies. Thank you all very much for listening, and I hope that there will be a good discussion.